Thank you for joining us today. This webinar will cover how to effectively reduce and eliminate FBS, human serum or human serum-derived components through the use of insulin, transferrin, and albumin supplements. These supplements are a great tool for both small-scale research and development, reduced serum cell culture, as well as large-scale serum-free manufacturing applications. This will be a 30-minute presentation followed by 15 minutes of questions and answers. If you have any questions during the course of the presentation, click the chat icon in the bottom left-hand corner of your screen and submit your question. I will be able to address your questions during the Q&A session. This webinar will be recorded and will be posted on our website for your reference. You'll receive a notification when the email is posted. Before getting into the content, here's a little information about myself. I joined Invitria in 2012, and I currently lead the product development team in both new recombinant protein development as well as the formulation of chemically defined, blood-free, complete media for end users in primary stem and virus production spaces. My background is in media and large-scale bioreactor optimization for monoclonal antibody production in CHO cells. Prior to that, I was doing immunotoxin research looking at engineering bacterial toxins such as diphtheria and anthrax to selectively target cancer cells, as well as the large-scale production of these immunotoxins in yeast and bacteria. So now that's enough about me. On to the reason why you have decided to attend this webinar. Fetal bovine serum, or FBS, was first utilized to propagate cells ex vivo in the late 1950s as described by Puck et al. This would start what would become a long-standing tradition in cell culture for years to come. FBS provides stimulation of cellular proliferation by the presence of growth factors, nutritional support through sugars, amino acids, and other undefined components, and the facilitation of cell adhesion via the extracellular matrix proteins. Over the years, FBS has proved to be a vital tool in cell biology as the versatility of the substance has enabled the establishment and subsequent study of several different critical in vitro cell culture systems, and as a result has greatly facilitated the biological research. Although there's broad adoption of FBS in both academia and industry due to its versatility and ease of use, the removal in both research and development as well as in the expansion of cells and cell-derived products for therapeutic applications has been recognized as a priority for a couple of different reasons. As you can see from the table presented here, limited supply is a big issue. In New Zealand, Australia, Mexico, and Canada, supply is dropping for a few reasons that are noted in the table. In New Zealand and Australia, the herd size is dropping. In Mexico and Canada, the increased demands of the U.S. are reducing the supply in these countries. In the U.S., the supply is actually expected to stabilize. However, in every country noted in this table, you can definitely see an increase in the cost of the supplement. This is presumably due to the overall decreasing supply globally. Further, what's also a bit disturbing about the data presented in this table is that as supply falls and costs rise, the abundance of or the absence of exact annual production volumes and loose regulations creates conditions to encourage unethical practices and even blatant misconduct of groups looking to maximize profit in this market. Indeed, alteration and fraud has been well noted in the serum industry even recently. In 1994, 30,000 liters of FBS were documented to be sourced from New Zealand. However, the country itself only reported 15,000 liters of annual production that year. A second major adulteration case in 2013 was documented with the dilution of FBS with bovine albumin, water, and other growth-promoting substances by a major global supplier. Subsequent investigations by the FDA concluded that approximately 280,000 liters of FBS were affected between 2008 and 2013.
So, serum supply aside, the addition of undefined, highly complex substance such as FBS to extremely sensitive in vitro systems that cell culture is absolutely presents another issue in terms of the potential for a large degree of variability, introducing a large degree of variability into the system. So this data presented here demonstrates the variable composition of different lots of FBS in terms of the average and then the range that's presented in brackets here. And as you can see, there's quite a wide variation in terms of the components within serum that can be found from lot to lot. So just to give you an example, progesterone has been noted to have approximately 120-fold variation between lots of serum. Very similarly, prostaglandin E has been noted to vary about 60-fold. Total protein, which includes albumin, insulin, any growth-promoting cytokines that are present in the lot of FBS, has been noted to increase to 2.5-fold. And so minus endotoxin, which is noted here, which can also vary 125-fold, and most cell culturists know that endotoxin is actually a very biologically active substance within cell culture and can really drive different responses within cells. Most components, endotoxin excluded, most components here are in some form or fashion included in different serum-free formulations, and so as you can see, the variation of these components can really drive variability in your desired application. So to kind of drive this point home, this is a study that's been published showing the effect of 4 different lots of serum in a standard cell culture model, MCF7. So some of you out there may have a lot of experience with this cell type. And this is basically showing 4 different lots of serum looking at growth kinetics, overall cell yield within 6 days of that growth kinetic study, and then the relative metabolic activity, morphology, and genotype, genetic expression of 6 different genes within these 4 different lots of FBS. And really the overall take-home message here is that there is a substantial difference between these lots of FBS. And honestly, this data is no surprise given the variability that I've noted in the table in the previous slide. So this data taken together is really, at least in part, the reason why there is a very well-known reproducibility crisis within the biomedical research field, in that there is a significant issue in the inability of researchers to reproduce published cell culture-based data. Okay, so given the issues with both supply as well as the reliability of data produced in FBS-containing media, the strategy of reducing the amount of incorporated into any given cell culture media makes sense, and the benefits of reduced serum cell culture can become very apparent once you actually look at the data. First and foremost, if you reduce the amount of FBS present in the cell culture media, what you're doing is you're actually reducing the variability of the system as a replacement of serum proteins with recombinant versions actually reduces the FBS dependency of that cell culture system and can actually enhance the reproducibility. Secondly, as mentioned, the price and availability of serum are really moving in an unfavorable direction for end users, whether it be for basic research or for manufacturing use. So including recombinant supplements that are aimed at reducing FBS inclusion can actually stretch your current FBS supply, which will do 2 things. First is that if your research is absolutely dependent on specific lots of FBS, you can certainly stretch that FBS, that current FBS supply and make it last longer. And secondly, it will absolutely make future serum purchases a little bit less painful in terms of financial implications. And finally, including supplements aimed at reducing the final serum inclusion level will actually save your lab some money in the long run overall. So just to kind of demonstrate this case, with serum prices increasing on a pretty consistent basis, typically the FBS cost in a typical liter of cell culture media with 10% inclusion will cost approximately between $80 and $117 per liter, depending on what source grade and vendor you're currently using. If you include supplements such as ITSE A, which I'll touch on more here in a minute, which can effectively reduce serum concentration by, absolute worst case 50%, but sometimes even you can reduce in specific cell lines up to 80%. But for the sake of this example, if you can reduce serum by 50% by including 5 mils per liter ITSE A, you can actually derive about a 30% cost savings per liter of growth media without sacrificing any performance of that media. And again, I'll touch a little bit more on this later on in the presentation. Okay. So as mentioned earlier in the presentation, FBS provides stimulation of cellular proliferation by the presence of growth factors, nutritional support, and facilitation of cell adhesion by the presence of extracellular matrix proteins. Some of the most prominent proteins in serum include insulin, albumin, and transferrin. So insulin is typically found between about 50 and 80 picomole per liter. Albumin is somewhere in the neighborhood of 35 to 50 grams per liter, and transferrin is between about 1.5 and 4 grams per liter. So if serum is reduced to, say, 50% of the original concentration, the major components can actually be held constant by the inclusion of recombinant versions. So Invitria currently offers several different blood-free recombinant insulin transferrin supplements in order to do just that. Together, these supplements represent the highest quality insulin transferrin supplements that are available on the market today for a couple of reasons. First, these supplements contain only recombinant proteins and do not incorporate any components from either human or bovine serum. There's quite a few different ITSE or ITS products that are currently marketed, but most, with the exception of Invitria's, actually incorporate human serum-derived transferrin. Further, these recombinant proteins are expressed in a non-mammalian host. Therefore, we do not use any CHO or HEK-derived recombinant proteins. So therefore, given these two quality attributes, we can consider these supplements to be totally blood-free. In addition, since these supplements contain only recombinant proteins, you can absolutely expect the highest degree of purity, quality, and reproducibility. And finally, these supplements are manufactured right here in the good old US of A in Junction City, Kansas. And Invitria owns and operates the entire supply chain related to the manufacture of these recombinant proteins that are included in these supplements. So currently, Invitria offers 3 different iterations of insulin transferrin supplements, ITSE, ITS, and ITSE Plus A. So each product is formulated as a 100x sterile liquid and contains recombinant insulin. And so at a 1x incorporation level, that comes out to be 10 mg per liter of insulin final. And as I mentioned, recombinant transferrin, which is actually our octaferrin product, at a final concentration of a 1x at 5.5 mg per liter. And these formulations are published on our website and are accessible at any point. So these formulations also contain sodium selenite, which is an extremely powerful antioxidant that's currently incorporated into most serum-free media formulations. And is, as I said, a very powerful antioxidant. So there's a couple of differences between these products that are worth mentioning. So the ITSE product, in addition to having insulin transferrin and selenium, also contains ethanolamine, which is an important lipid precursor that's required for some cell types, but not for others. The ITS product just, again, contains only insulin transferrin and selenium. And then ITSE Plus A is our ITSE product, so it contains selenium and ethanolamine, in addition to our recombinant albumin, cell culture grade albumin known as Celistim S, at a final concentration of 200 mg per liter at a 1x incorporation level. So now that I've introduced these 3 insulin transferrin supplements, first I want to give you a few examples as to how these products can be utilized in first reduced serum culture, and then we'll move into the more sophisticated serum-free media formulation. So comparing the 3 products together, really ITSE Plus A is the most well-suited for reduced serum cell culture, as this supplement includes the recombinant albumin. So albumin has multiple roles within cell culture media, and so it is extremely effective, and you will see a benefit or additional benefit from including this component in reduced serum conditions. So you can gain the same effect by using ITSE or ITS with additional Celistim S supplementation. But for the sake of simplicity, the following reduced serum cell culture examples will be performed with ITSE Plus A. So in this particular example, this is an SP20 hybridoma, murine hybridoma line that has been traditionally grown in DMEMF12 with Glutamax, the glutamine dipeptide, in addition to 1X-F68 10 millimole heaps and either 10% FBS, 2% FBS, or 2% FBS with the final inclusion of 8 mil per liter ITSE A. So in order to generate this data, cells were passaged for 3 serial passages in dynamic shaking culture, and the doubling times of each passage were averaged together to present the data shown here. So as you can see, the inclusion of 8 mil per liter ITSE A with 2% FBS is actually able to rescue these cells in reduced serum culture versus 2% FBS alone. Further IgG quantification, which I'm not showing here, but IgG production was also restored by the inclusion of ITSE A. Okay. Here's another example, but instead of suspension culture, this is an adherent cell type. So HT1080 is a fibrosarcoma cell line that's traditionally used for cancer research. And so HT1080 cells are usually cultured in EMEM. And so here the full growth media is an EMEM with 2 millimole glutamine and 10% FBS. And so we were able to essentially rescue, if you reduce serum by half into a 5% inclusion rate, you actually see a pretty significant increase in the doubling time of these cells. But with the inclusion of 5 mil per liter ITSE A, you're able to restore the doubling time back to be equivalent with 10% FBS. So the other thing to mention here is that morphology as well as other characteristics that you would expect of a tumor cell line were not at all altered by the inclusion of 5 mil per liter ITSE A. Okay, so the 2 previous examples gave a proof of principle that you can effectively reduce FBS using ITSE A. So this type of approach is relatively straightforward in that really the only optimization work that needs to be done is identifying the lowest level of FBS inclusion that your cell type will tolerate and still be able to be rescued using the ITSE A supplement. There is a potential pitfall in that if you reduce FBS too far, that the supplement cannot rescue the reduced FBS culture. So this would indicate that there are other proteins or components present within serum that have been effectively reduced to levels that are below what would be required for that cell type, and that replacing only albumin, transferrin, and insulin are not adequate to rescue that concentration of serum. So, however, in this type of approach, what you're doing is you're really relying on additional components that FBS is providing in order to support your cell culture system, and you're only adding back a few components within the media. So it's a pretty simplistic approach, but very, very quick and easy to do. However, if you're looking to completely remove FBS from your cell culture media and formulate your own custom blood-free chemically defined formulation, there are absolutely a few advantages over to the reduced serum approach in addition to the benefits that I've outlined earlier with the reduced serum. First and foremost, you're actually removing all undefined components from the cell culture system and replacing it with a media that is completely chemically defined. And what I mean by chemically defined is all of the components within that formulation are known. So this can actually, this statement can actually lead to a couple of 3 key advantages of a totally chemically defined media over just a reduced serum culture. So first, during the development process, identification of media components that are critical for the function of your cell product are actually an extremely rich source of patents that can further enhance your company's position within the marketplace. Secondly, definition of the media system can identify avenues for cell product potency and functional improvement. And finally, if you are able to develop your own media formulation, you can actually avoid media formulation disclosure issues by large media suppliers and potentially facilitate the regulatory approval process. Okay. So although there's some clear advantages to going completely serum-free over, say, reduced serum cell culture, there's some obvious hurdles as well. So primarily, serum-free media formulation is a somewhat complex, and the time investment is much more demanding to develop a totally chemically defined serum-free media formulation than it does to just simply reduce the serum in your cell culture. So there's a lot of other considerations that you have to look at. Simply adding ITSE A into your current base media that you use to supplement with serum just simply won't cut it. You'll actually have to consider several other additional compartments within the media and optimize each of these, such as your antioxidant and polyamine trace element profile, your amino acid supplementation, your water-soluble vitamin and lipid profiles. Additionally, you'll have to identify the correct cytokine and recombinant protein supplementation, such as insulin, transferrin, and albumin as well. So finally, you'll also have to consider the optimal pH, media osmolarity, as well as ideal buffering systems in serum-free media. So some of these topics are going to be outside of the scope of this webinar today, and we won't really have much time to touch on a lot of these points. However, what I'm hoping to do today is give you some examples of cell types that we have optimized completely blood-free and chemically defined media for, and how the blood-free supplements that we've just gotten done talking about, the ITSE and ITSE A, have been used in these formulations and other in vitro products that are needed to formulate compartments within these complete media formulations. So we actually utilize a DOE-based statistical platform in order to optimize media formulations. But here's a general workflow of how we go about developing a blood-free chemically defined media. Now, I must warn you that just the simple substitution of a recombinant protein into formulations that are actually built around serum-derived albumin and transferrin is an approach that will not be successful. Serum-derived components actually carry significant biologically active contaminants that when you remove these components from media formulation, you actually have a formulation that is actually inadequate in order to support sufficient cell proliferation and phenotype retention in the presence of recombinant alternatives. So at Invitria, the way we actually start with blood-free media design is to select the albumin and insulin transferrin supplements first. After these supplements have been made, we actually begin the formulation work by building the formulation around these products using DOE designs. So what these components are that are being considered, that are being built around the Invitria supplements are, of course, antioxidants, polyamines, certain trace element cocktails, and cell type specific cytokines. So for instance, in the case of MSC, you would be screening these components in the presence of basic FGF, TGF-beta, and PDGF. Okay, so once we come up with the optimal supplementation profile, we then begin to optimize these reagents in the presence of the Invitria supplements. And then once we think that we have an optimized reagent profile, then we actually optimize the Invitria supplements, holding the previously optimized supplements as constant. This overall will generate the very rough framework of the formulation, and then we move into subsequent optimization schemes to look at amino acid profile, and then finally look at osmolarity as well as buffering systems. Okay. So once a formulation or formulation is identified as being optimal, we will actually scale these formulations up into multi-passage tests that will confirm cell functionality as well as desired phenotype. So here's an example of a blood-free T lymphocyte expansion media that was formulated in my lab for a CAR T therapy. So we actually have quite extensive experience in terms of knowing the optimal concentrations of different components in different cell types. And so we know the optimal concentration for T cells for ethanolamine and selenium insulin transferrin albumin are within these ranges. And so since we know these ranges, really what we would like to do is figure out what in vitro products would work the best in this application. And so looking at the formulations of ITSE, we actually found that using a 2 to 4x ITSE concentration will bring most of these reagents into the ideal concentration ranges. Alternatively, you could use the ITS product at a 3x final inclusion level in order to separate the ethanolamine inclusion to see if the higher inclusion from a 4x ITSE would actually be detrimental. Further, we do know that the expansion of human T lymphocytes is absolutely dependent on the presence of albumin. These cells will not expand in the absence of albumin. And we know that T cells like to be somewhere in the neighborhood of 500 to 1,000 mg per liter of albumin. So we actually included 1,000 mg per liter of albumin in this formulation. So what we did finally decided on a 2x ITSE with 1,000 mg per liter of cell-MS. And here's the results. So these are healthy T cells that were essentially expanded out of a PBMC pellet that was from whole blood from healthy donors using just a Ficol gradient, and then there was no further selection. They were activated with CD3, CD28 dynabeads and expanded for 7 days in either a commercially available xeno-free, serum-free media that contains human serum-derived albumin and transferrin, or the chemically defined blood-free media from Invitria. So as you can see, there's a very nice increase in the potential for cell growth. And in terms of the desired phenotype, we actually found that these cells, the client actually had a very specific requirement of these T cells in terms of their phenotype. They knew which phenotype was driving potency of their cell product. And so as you can see, we actually saw a very nice increase in the percent of the desired phenotype that was coming out of that expansion process. Okay, so here's another example as to how some of these blood-free supplements can be included in complete blood-free media formulations. So this is a virus production media that's been optimized specifically for Vero cells. So Vero cells are an extremely popular continuous cell line for virus production. They're very easy to handle. They grow well. These cells grow readily in EMEM or DMEM plus 10% FBS, depending on which paper you're reading. They've been very well accepted by the NIH and WHO for vaccine manufacturing. And so in an academic setting, the way, at least in serum for virus production, what people do is typically grow these cells in 10% FBS, and then during the virus production phase actually remove the FBS and just make the virus in either a basal serum-free media, or they lower the serum to 1 to 2%. So again, my lab has extensively characterized the optimal concentration ranges of ethanolamine, sodium selenite, insulin transferrin, and albumin for Vero cells. And so these optimal concentration ranges are given here. So again, we use this information to build a blood-free chemically defined media around these reagents. Given the optimal concentration ranges of each of these components for Vero cells, we decided to actually incorporate the ITS product instead of ITSE. So The reason for this is actually Vero cells prefer very high levels of ethanolamine. Since ethanolamine is only present at 2 mg per liter final with ITSE as a 1X, a really high inclusion level of ITSE would be required. This would actually cause an excessive amount of insulin transferrin and sodium selenite, which would actually be very detrimental to Vero cells. So as you can see, the transferrin level is actually quite low for Vero cells, and the data would indicate that concentrations over about 15 to 20 mg per liter of transferrin is actually quite detrimental to these cells. Another issue is sodium selenite. Again, these cells are very, very sensitive to higher levels of that antioxidant. So basically, the other really important thing to note here is that Vero is not at all as dependent on the presence of albumin as, say, the T lymphocyte example that I gave in the previous example, or other cell types such as MSC. And so, to be quite honest, they just don't require as much. So you'll see that the albumin level, recommended albumin inclusion level, is actually quite low. So we found that selustim-S did very well for this cell type, and decided to include it in the final formulation. So again, the final ITSE supplement profile was a 1X ITSE, ITS plus selustim-S, and then we just supplemented additional ethanolamine into the formulation to get that higher level of ethanolamine that Vero cells prefer. So we took this supplement profile through the DOE platform, and this is what came out on the other side. So this is actually our OptiVero product. So this media is actually commercially available for sale. And you can see this is a 15-passage T-flask experiment that shows the expansion of the blood-free chemically defined OptiVero media versus EMEM and 10% FBS in wild-type Vero cells. Further, we did test the performance of this media in the expansion of dengue virus, Zika virus, as well as 4 different types of influenza virus. And we found that the media was actually very able and efficient to expand all 4 virus types, and actually even better than the commercial media VPSFM, which was found to have some pretty significant issues in some of these virus types. If you want to see the full study for this particular media, I would invite you to actually download our poster that was presented at the World Vaccine Congress in Washington, D.C. in April of this year. If you'd like the link for that, Chuck can absolutely help you find that content. Okay, so to bring this webinar to a conclusion, I'd just like to again summarize how Invitria can help facilitate both your research as well as for those of you working in industry applications looking at commercializing either cell or a cell-based product. Invitria produces and markets a portfolio of blood-free components, supplements, and complete media. And again, just to reiterate, what we mean by blood-free is that these components and supplements are not sourced from either human or bovine serum. There's a lot of confusion that's currently in the market with xeno-free, animal-free. So, we've coined this term blood-free, and we believe that this is the absolute highest quality and highest level of animal-free that you can be. Further, these recombinant proteins are not manufactured by a mammalian host such as CHO or HEK. These recombinant proteins are highly purified, well characterized, and as you can see from the previous data, extremely biologically active. So as a result, these proteins have exceptional reproducibility in cell culture systems. Further, my group, through the years, we've developed a deep expertise in the application of these proteins in industry-relevant cell types. To date, we have formulated proprietary media for end users using these components and supplements for Vero cells and T cells, which you've seen here, HEK293, MSC, ARPE19s, A549s, Tregs, iPSC, ESCs, fibroblasts, and neural stem cells. So as a result, both the blood-free supplements such as ITS, ITSE, and ITSE A as well as complete media formulations designed by my group, have been implemented in commercial manufacturing processes for stem cells, vaccine, virus, and tissue storage applications. So they've been well utilized in industry, even with some products moving into BLA and commercial phase manufacturing. We routinely share the knowledge that we have gained with groups interested in removing all blood-derived components, whether it be FBS, human serum, or components purified from human serum from their manufacturing processes. We believe that the use of blood-derived components, although they've had their use, we feel that now with this enabling technology that there is a better way. We actually share this knowledge in a few different collaborative formats, and we try to be as flexible as possible for the end user. If you would like more information as to how we can help you remove blood-derived components from your research and manufacturing processes, you can absolutely reach out to one of our product application specialists, which I will show you. So really, these are kind of our experts in different areas of the Invitria business and research and development. So Scott Dieter, who's on our far left, is our CEO. Vladimir Koyev is our VP of manufacturing. He's in charge of all of the manufacturing activities in our Junction City facility. Myself, I'm in the center, and again, I'm in charge of most of the applications as well as the product development and new R&D. Ron Backman, who's our VP of Quality, who's based out of Junction City, Kansas, has been absolutely instrumental in helping customers in CMC-related issues and bringing these use of these products to the regulatory agencies. And then Addie Pennybaker, who is one of our product application specialists. We actually just had another addition. Marcus Hoffer, who is also on the applications team. Either one of these product application specialists can answer any questions that you have around optimal inclusion levels for different cell types or other components and supplements that we would offer. So if this webinar was convincing enough and you feel like you're ready to evaluate one of these blood-free supplements that we've discussed today, there's a couple of ways that you can order. You can go directly to our website, create an account, and purchase directly online, either with a PO or a credit card. If you have any questions, please feel free to reach out to our product applications team at these phone number or email address below, and we always try to be as prompt as possible in getting back to you. So again, thank you for your time today. I hope that you were able to take away some valuable information from this webinar. In closing, we feel that the removal of blood-derived components from all manufacturing processes is absolutely vital in ensuring the safety, reliability, and ultimately the success of novel life-saving cell-based therapeutics. And so we feel very passionately about this. And so if you have any questions, again, please don't hesitate to reach out to Invitria's technical team, and we'll be there to answer any questions you may have. And again, thank you for your time today.
Great. Thank you very much, Randy. We have several questions in the queue. So the plan is to spend the next 15 minutes or so going over some of the questions. For the people that are still going to stay on for the Q&A, there's still time to submit your questions. Please do so in the bottom left-hand corner. Click on the question icon and enter your questions there. So Randy, the first question we have for you is, what product do you recommend for cell line development and seed train expansion in CHO cells?
Yes, that's a good question. So I would honestly, so I'm assuming that these would be suspension CHO, and so I would honestly start with something like Joclix EMEM. The reason for this is it really is a light media, and it really gives you a solid platform to work on optimizing amino acid profiles as well as other reagents that are needed. The advantage to using Joclix EMEM is it's basically EMEM minus calcium and magnesium. And so you absolutely do not want these two ions in your suspension culture. If these are adherent CHO cells, I would highly recommend DMEM F12 for most adherent cell types. We start our development with DMEM F12 just because it is a highly fortified media, and of course it's further manipulated down the line, but it's a really good starting point for most adherent SFMs.
Excellent. Thank you, Randy. The next question here is a three-part question. So what level of animal-free are these components? That's part one. And then the second part is the host animal-free? And thirdly, what about processing packaging components?
Yeah, absolutely. That's a great question. So, and this is kind of going back to the comment I made earlier. We feel that there is a lot of confusion in the marketplace. Xeno-free, animal-free, so on. What does it all mean? So we've actually coined the term blood-free, which is actually synonymous with a tertiary level of animal component-free. And what that means is that the expression host is non-mammalian, non-animal. So this is actually a non-mammalian-based expression platform. Further, the facility is considered to be animal-free. So what that means is all of the vital equipment that comes in contact with the product has never seen animal components, nor has any of the critical processing instrumentation. And then finally, the packaging that holds the final product is also certified animal-free. So as I said before, we've tried to come up with a term that really encompasses this tertiary level animal component free idea, and we feel that is blood free. And so I probably should have elaborated a little bit more during the webinar, but that is, we believe, the highest level of animal component free you can be is animal component free to the tertiary level.
Great, thank you, Randy. The next question is, what base medium would you use to test concentrations of ITS or ITSE for suspension culture of HEK 293?
Yeah, that's a great question. So again, I would either evaluate a media that's been formulated for suspension culture. So that's either going to be RPMI 1640 or again the Joclix EMEM. So really, again, the issue is that in suspension culture, you need to make sure that you don't have high enough levels of the calcium and magnesium ions to interfere. If these are present, you're going to see a lot of cell clumping, and it's going to make interpretation of the data extremely difficult. It's very hard to count clumps of cells.
Excellent. All right, so here the next question is, are customers able to outsource the formulation of blood-free media for specific cell types to Invitria?
Yeah, absolutely. So as I mentioned, we work with customers in a very collaborative, transparent fashion. We actually very recently launched a media formulation service where you're able to partake in the development of your own custom media formulation. If you have further questions around how this process works, please feel free to reach out to our product applications team. They can get you all the details.
Great, thank you very much. Next question is the transferrin and insulin included in the supplements recombinant?
Yeah, so that's a little bit confusing sometimes when people talk about ITSE and ITS and ITSE plus A. What does it mean? I mean, the name of the supplement essentially tells you what's in the supplement. So ITSE is insulin, transferrin, selenium, ethanolamine. ITSE plus A is ITSE plus albumin. So there's also a lot of confusion in the marketplace. Which one do I need? And what is, like, I know Sigma has a line, insulin plus +1, +2, +3, you know. So we try and make it as simple as possible. But again, if you have any questions about what ideal product would be for your particular application, please feel free to reach out to us, and we'll be there to help you out.
Great, thank you. Next question is, this person wants to know about how Invitria works with end users to formulate custom media.
Yeah, so we do that in actually two different ways. And it's kind of dependent on how the end user would like to engage in Vitria and really leverage the expertise that we have. If they want to be very involved with the development process, what we do is we actually send the DOE platform to them, and they run all the experiments. And then they send us the data back for evaluation of the data and then formulation of the next round of media candidates. Alternatively, if you just don't have the manpower and the resources to do the work yourself, Invitria can actually do the work for you, where we do all the formulation work and then we provide you with candidates. And when I say candidates, I mean 3 or 4, not 25, for you to evaluate, and then give your input on the formulation, and we'll make the corrections for any issues that you have seen, and then send you back the final formulation.
Great, thanks, Randy. 3 more questions left here in the queue. Do you recommend 10 milliliters of ITSE per liter of final media? That's part 1. And part 2 is how much insulin and transferrin is in 10 milliliters of ITSE?
Yeah, so that's a really great question. Just because it is designated as 100X doesn't mean you have to use it as a 1X. And the T-cell example is a really great example. We know that, T lymphocytes, they're actually a little bit of transferrin hog, and honestly, our experience has been that you want to bring that transferrin up to between 30 and 50 mg per liter. At a 1X concentration of ITSE, it's only at 5.5 mg per liter, and you're absolutely going to see the difference, in terms of the expansion capabilities, especially in more confluent cultures. I mean, T cells have a very robust proliferation response, and so, confluent cultures, cultures that are over 1e to the 6 cells per mil, it's going to get, you're going to deplete the transferrin in the media very, very quickly. Or at least, have more cells than available transferrin molecules. So just because it is designated as 100X doesn't mean that it's a solid 1X. So, you can use more, and in fact, in the case of CHO, again, that's another cell type that's very, very hungry for transferrin and insulin. We would actually recommend, 2 to 3X a final inclusion. To answer your question about the final formulation, I believe at a 1X insulin is going to be 10 mg per liter. So I'm doing the math in my head, and again, I apologize if I mess this calculation up, but I believe it's 1 gram. The formulations are actually published on our website, and they're available for your reference.
Great, great. All right, 2 more questions. This next one is more of a customer service question. How can I get technical support for using ITS plus albumin by email?
Yeah, so that's a great question. You can either just go directly to the website and hit the contact us, or you can just use this email address here, info@invitria.com. And that email will be triaged to the correct person, and we usually try and respond within 24 hours.
Great, thank you, Randy. And the last question is just again for customers interested in ordering. What is the best way to order products through Invitria?
Yeah, so we try and make this as easy as possible. I would encourage everybody here to go onto the website and make an account with Invitria and just submit your orders online. Usually they ship either same day or next day.
Excellent. Well, thank you again, Randy. No other questions in the queue, So that pretty much wraps up today's webinar. Again, what we'll be doing is sharing this recording and putting it on our website, as well as emailing everyone that attended today links to the recording. Feel free to reach out if you have any questions, and thanks again for joining today's webinar.